Here we go with part 2 for protein synthesis. So the steps in translation, there are 3 of them. They are pretty straightforward, I think. The first step is called initiation. And if you think about it, to initiate something is to start something. So this is the start of transcription. And remember that transcription is taking a DNA template, a DNA model, and converting it into RNA so that it can leave the nucleus, because DNA is too big to leave the nucleus. RNA is single-stranded, so it can go through the nuclear pores and leave the nucleus to get to the ribosomes, which are in the cytoplasm. There are a couple parts to the initiation part of transcription. The first part being that there are these proteins called transcription factors. You're not going to have to know what the names of them are, what they look like, what order they attach, just transcription factors bind to or attach to something called the TATA box in the promoter region of the gene. Now, we said in the first video that the promoter region is the place where the transcription factors and the RNA polymerase are going to attach. And go ahead and get your giggles out over the TATA box. It has nothing to do with that kind of TATA. It is thymine, adenine, thymine, adenine. So get your mind out of the gutter. So after the transcription factors bind the TATA box, then the RNA polymerase II can attach to the transcription factors. Very straightforward. What I have on here, this picture is not the best, but that's why I added these brackets in. Here's the promoter region with the TATA box, and the TATA box is actually on the non-transcribed strand of DNA, so it's on the 5' to 3' strand of DNA, but that's kind of off on a tangent. You don't really need to worry about that too much. But this is one of the transcription factors. The next picture I have is going to be showing all of the other transcription factors that attach. The transcription unit, or the genes, the bases of DNA that are actually going to end up becoming the protein, are in this transcription unit. If you remember when we did the trip operon, there was also a transcription unit to each of the operons. So here they're showing, let's see, 1, 2, 3 transcription factors. I think there are more transcription factors than that, but I don't really care because we don't have to know it for AP Bio. These transcription factors attach first, then the RNA polymerase can attach, and that's the end of initiation. Transcription factors attach to the TATA box, RNA polymerase attaches to the transcription factors. Easy peasy lemon squeezy. So the next part in the process is elongation, and it is exactly what it sounds like. It's the RNA that's being produced, and it does not matter whether it is messenger RNA, transfer RNA, or ribosomal RNA, but the RNA that's being produced gets longer. In other words, the RNA polymerase travels through the transcription unit, adding RNA nucleotides to the 3' to 5' strand of DNA. And the order in which the RNA nucleotides is added is not random. That's why it makes a difference whether you're using the 3' to 5' strand or the 5' to 3' strand. We are still going to follow the rules of complementary base pairing. The only difference is that instead of adenine bonding with or pairing with thymine, adenine is going to pair with uracil because uracil replaces thymine in RNA. So what this is showing, in part A, this is showing at the end of the initiation. And this is not the best diagram, but I liked that it was very simple and very straightforward. So this yellow section here would be the transcription factors that have attached to the green promoter region, and then this light blue RNA polymerase II attaches to the transcription factors. And then when you see that whole complex right here, it is separating this 2 strands of DNA and starting to make the messenger RNA. This darker blue, this medium blue arrow is showing the messenger RNA that's being produced. So the RNA polymerase is moving in this direction from left to right. If I can get this to actually work. There we go. RNA polymerase is moving left to right, And as it transcribes the 3' to 5' strand, see down here it says 3', down here it says 5'. Remember, 3' has the deoxyribose on the end, 5' has the phosphate. As that happens, the RNA strand that's being produced starts getting longer and longer and longer, hence elongation. So the transcription unit is this dark yellow, and as the RNA polymerase goes through the DNA from the beginning of the transcription unit to the end, this medium colored blue RNA strand is produced. The last step then is termination. And as I'm sure you know, to terminate something means to end it. And the ending or the termination of RNA polymerase and this whole transcription process is pretty straightforward. The RNA polymerase 2 reaches a sequence of bases that does not physically allow the RNA polymerase to go any further. It's almost like there's a roadblock, like concrete blocks that are in the way. And these concrete blocks, as the RNA polymerase is coming down the DNA molecule from 3' to 5', what happens is that the RNA polymerase and the transcription factors run into this termination sequence and just go boom, and they fly apart. Just think about what would happen if you ran your car full speed into a concrete wall. A really thick concrete wall that's reinforced with rebar and all wonderful things like that. Your car would fly apart into pieces. Then, because the DNA was never completely separated from the two strands from each other, the DNA can just snap back together because those hydrogen bonds that hold the DNA together are weak and easily broken, but they are also very easily reformed. And your RNA strand can float away and go out to the cytoplasm to be translated, if that's what it's supposed to do. Or it can go to the nucleolus to be combined into making a ribosome. I am not going to click on this link. I don't think I can in this version of the screencast that I'm using. Maybe I can, maybe I can't, but I'm not going to. You guys can go ahead and do that on your own. And that should take you to a very nice video on transcription. But we're not quite done yet with transcription. Here's a picture, first of all, of termination. The red part here is the terminator region. Makes me think of Arnold Schwarzenegger, the Terminator. And the RNA polymerase and the transcription factors literally fly apart, which releases the RNA strand. All right, so before RNA can actually be used in translation, it has to be modified. And there are a couple things that have to happen. First of all, on the 5' end of the RNA molecule that was produced, a cap needs to be added. And this cap it's a guanine nucleotide that's just been modified. And you don't have to worry about what type of modification that it has undergone. So there's a 5' guanine cap. And on the 3' end, there is added what's called a poly A tail. And the A in this case does indeed stand for adenine. So poly means many. A stands for adenine. So many adenines. In fact, anywhere from 50 to 250 adenines are added to the 3' end. Oh, but wait, that's not all. We have to talk about introns and exons for a couple minutes. You see, in that transcription unit, not everything that is transcribed actually makes it to the next step to be translated. There's some extraneous material that needs to be cut out. Unfortunately, for me anyway, it always seems backwards, because to me, exon means like you X out, you get rid of this part, but it's the opposite. So the exons are the good part. The exons are the part that you're going to keep. The exons are the part of the RNA that are expressed and are going to become part of the protein during translation. Introns do not become part of the protein. They are useless. So the introns get in the way and the exons are expressed. So exons are expressed, introns get in the way. So we want to get rid of the introns before translation takes place. The way that that happens, there is an enzyme called spliceosome, which really freaks me out because, first of all, it doesn't end in ase, and second of all, that sounds like some sort of organelle or something. You know, we've got lysosomes and we've got ribosomes, and no, this is an enzyme, and this enzyme is used to cut the introns, which get in the way, out of the RNA. Here's a picture. So in this picture, the exons, remember the exons are the good ones, so the red and the yellow you want to keep. The blue is the part that you're going to get rid of. But in the process of transcription, none of that matters. This whole gene, this whole transcription unit is going to be transcribed. So your RNA that's produced as a result is going to have, look, red, yellow, blue, yellow, blue, yellow, blue, yellow, red. While in this RNA that's produced, you've got red, yellow, blue, yellow, blue, yellow, blue, yellow, red. It's the same as the DNA. But then, when you go through this splicing with spliceosome, what you end up with then is intron 1, intron 2, intron 3, intron 4, and the exons are gone. And then as it goes through, this would be protein synthesis, this would be translation. This is representing, each of those balls represents an amino acid that becomes part of the string. So that is the process of removing the introns, because the introns get in the way, using spliceosome. So our next segment is going to be starting with translation, and I will do that in a separate video.